Good morning, everyone. For those of you that don't know me, my name's Fiona Angrisano, and I'm a Senior Research Officer within the Vector-Borne Disease and Tropical Public Health Working Group at the Burnett Institute. It's my pleasure to talk to you this morning as part of this Shiny workshop. And I'm really excited to introduce the Luminex technology and talk to you a little bit about how we go about setting up a serological assay. So I'd like to start firstly with talking about XMAP or multiplexing technology. XMAP technology allows users to perform a wide range of protein and nucleic acid-based multiplex assays. And these assays can simultaneously detect up to 500 targets in a single run, depending on which machine we're using. So the advantages of this technology are that we can test for more biomarkers, say, than your standard ELISA, which looks at one antigen. We can develop custom assays, so we can really tailor the assay to specific questions in mind. We can use a lot less sample than it's often required for other assays, which saves on time and reagents. And all of this together really helps us gain a better understanding of complex biological systems. So what is multiplexing? It's a method for high volume biomarker testing or testing multiple analytes simultaneously within a single run using a single sample volume. And XMAP technology is really best suited for this kind of testing, as its preferred range of targets is 3 to 500. So how does this technology actually work? XMAP technology uses labeled microspheres, or you'll often hear us refer to them as beads, and it allows for the simultaneous capture of multiple analytes from a single reaction. Because of their small size and low density, microsphere-based assays exhibit what's called a virtual solution phase kinetics. And this means that the beads are individually read using by an XMAP machine. XMAP actually stands for multi-analyte profiling, where the X represents the biomarker being tested. So this could be a protein, a nucleic acid, or a polysaccharide. And XMAP beads come in a variety of formats, including magnetic and non-magnetic. As these XMAP beads pass through a red laser, or LED, it excites the internal dye to distinguish the microsphere set. And then a green laser or LED excites the fluorescent reporter dye to determine the result of the assay. So that statement's presented here in this picture below, where a bead is hit by 2 lasers at the same time, a red one, which tells the machine which bead region it is, and then the green one, which actually excites the reporter and takes a reading for how much of something is present. Of the many machines that are on the market, the Magpix, which some of you will be familiar with, is the simplest and the most affordable of the Luminex machines. It's a compact multiplexing unit, which can perform up to 50 different tests in a single reaction volume, and it reads a 96-well plate in just 60 minutes. So we really like the Magpix, and we use it quite routinely at Weehai and Burnett, and there's one at POM and in Goroka. And the reason we like it is for a few key features that it has. So as I said, it multiplexes up to 50 analytes. So it's not quite at the 500 end or 500 analyte end as some instruments, but 50 is often more than enough for what we want to look at. It's really quite sensitive, so it can detect approximately 10 to the 6 copies of DNA or a single digit picogram level of protein. The antigen selection can be really flexible, and the format of the panel is also flexible. It's got a quick read time. So as I said, 96 well played in under 60 minutes. And if we're looking at that in terms of tests per hour, this machine could do 4,800 tests per hour. Comparing it to some of the other machines on the market, it's got a relatively simple and quick startup and shutdown routine, just under 15 minutes. And again, compared to other machines on the market, it's relatively small in terms of footprint. So it's only 3 feet in linear bench space. However, I know a lot of us are really tight on space in the lab, so that sounds like a lot. But when you're comparing it to some of the massive machines that are available, it really is quite compact for this kind of technology. I just want to move on to actually talking about the assay or the protocol that measures antibodies, specifically our COVID protocol, which has a number of steps involved. But I wanted to highlight firstly that this image is taken from a paper that's referenced below. And if you are interested, I'd encourage you all to have a look at this paper. It's by the team at WeHi, and it really is a method paper for setting up serological assays, in particular the COVID panel. So to sort of summarize, our Luminex protocol involves several steps. The first one is to couple our antigens to our carboxylated beads, and then to evaluate these beads using either plasma, serum, saliva, or monoclonal antibodies. The third step is to add our PE conjugated secondary antibody, which we often refer to as our detector antibody. Then we place the plate on the Luminex instrument for analysis, and we get a readout in terms of the number of MFIs detected for each bead region in the assay. So I'd like to take the next couple of slides to go through each of these steps in a bit more detail. The first being the bead coupling. So the optimal protein amount to couple to a magnetic bead has to be first determined, particularly for proteins that are in-house and not commercially made. So all proteins that we receive or decide to look at are initially coupled at 6.25 times 10 to the 5 pre-activated beads. And we couple at a variety of concentrations, so usually about 1 to 10 micrograms. And we refer to this as our trial coupling. The optimum antigen concentration is then determined by observing what concentration of protein resulted in a log linear standard curve using convalescent plasma. So this is really important in achieving an optimal dynamic range to be able to measure a wide range of different concentrations whilst measuring all of the samples at one dilution. For example, our plasma samples are always run at 1 in 100, and we really need the same beads to be able to detect any antibody responses that are quite high and some low responders as well. So that's why we often spend quite a bit of time during the beginning of a project to determine the optimum bead coupling ratio. Once determined, we perform a bulk coupling. This is just a matter of increasing the number of beads up to 2.5 times 10 to the 6. And when we've performed a bulk coupling, we can use that bulk coupling for up to 3,000 samples. Now, when we did this for a number of SARS-CoV-2 antigens, as well as other antigens in human plasma, such as respiratory viruses and human seasonal coronaviruses, these are the standard curves that we ended up with after optimizing the concentration of protein. So these graphs, A represents the IgG for SARS-CoV-2 antigens, and B represents the non-SARS-CoV-2 antigens. And each line is depicting the level of IgG antibodies that are present at a particular dilution. And these are represented as mean fluorescent intensity values. So along our x-axis, we have our serodilution or our standard curve, and on our y-axis, we have our MFI. And we really like our first dilution, our 1 in 50 or our S1, to be up in the high thousands to create a really nice standard curve. These particular standard curves were created using plasma that consists of convalescent plasma from 10 SARS-CoV-2 qPCR positive individuals. And this plasma was collected approximately 30 days after onset of symptoms. So we tried a variety of different antigens and played around quite a bit in terms of what was going to be the best collection of COVID and non-COVID antigens to answer some COVID sero survey questions. And the panel we finally ended up with is here in this table. And it gives us a mixture of both SARS-CoV-2 antigens and seasonal coronavirus antigens, along with a flu influenza antigen and a control of tetanus toxoid. You can read more about these antigens in the paper I mentioned, but essentially we settled on a combination that would allow us to detect sort of normal flu levels that are in a population, as well as SARS-CoV-2. But more importantly, the antigens we selected for SARS-CoV-2 really allow us to tease apart whether someone has antibodies because of a natural infection or due to an immunization. And so once you decide on your panel and you coupled all of your antigens and you have a nice log linear curve for each one, you can actually start thinking about running an assay. And so we will use our beads either with plasma, saliva, or monoclonal antibodies, and we set up our plate. How you label your plate for your own reference will probably be quite different to how the plate actually needs to be labeled to use the R-Shiny app. So some things to consider, which I guess is just good practice to get into when you're setting up a cohort, is the antigen nomenclature that you're going to use. Regardless of what you decide on, just be consistent throughout the study. Often the R-Shiny app doesn't like spaces in between its names, so you can use a dash line, but I wouldn't spend a lot of time going crazy in detail with the antigen name, but just be really consistent throughout. Again, with the bead IDs that you choose for each of your antigens, try and stay consistent throughout the study. So you might require a couple of bulk couplings for your cohort. And if you're not making it all up at the start, just with each bulk coupling you do, make sure the same antigen is coupled to the same bead region. If the study allows you and you know how many samples you're going to run, which I know can be hard because sometimes we have these ongoing cohorts, if you know the study size, my advice would be to make up enough bead antigen coupling at the beginning, so it just avoids batch variation between couplings. When you set up your plate, we recommend that you run 2 blanks, a negative control, and a positive serial dilution or your standard curve. And as I mentioned before, your standard can be convalescent plasma, as is used in the COVID assay, but it also can be a research reagent or antibody that you know is a known positive. The actual IgG antibody protocol, it has a number of steps. So you add your beads conjugated to antigen into a well, and then you add your samples. So your unknown samples or your cohort samples are added in singlet at 1 in 100 for plasma. And then you add your standard curve in the top row, and that's added as a serial dilution. The plate then gets incubated at room temperature in the dark because our microspheres are light sensitive. And then after a period of time, you wash any unbound antibodies. And our assay differs from the one depicted in this picture in terms of step 3 and 4 in our assay is combined. And that's because our detector antibody is already coupled to a reporter dye, which is great because it saves us a bit more time. So we add our detector antibody coupled reporter dye, and then we incubate again at room temperature in the dark. After a period of time, we wash any unbound antibodies and we resuspend our complex that's left in the well, ready to run on our Magpix. Prior to putting your plate into the machine, and often we'll do this during an incubation time, you need to ensure that the machine is on and primed for running. And that involves, again, a number of steps. So you have to run your daily startup protocol and then an enhanced startup protocol. We often adjust the probe height, and this is to ensure that the probe in the machine is not going to punch a hole in your well, but also not be too high up out of the sample. You want to calibrate the system with the calibration kit to make sure everything is in order, and then verify that the system's actually working with a verification kit. You want to make sure you've got enough wash buffer to run the assay and that the waste levels are empty. So once we've done all of that, we pop our plate onto the machine and we can go about setting up the protocol. And one of the first things that you'll get to is you'll get to select the bead ID regions. And these are represented as a number of circles with a number in them on the machine. And it's just a matter of going through and clicking which bead region you have and telling the machine that yes, bead region 12 is tetanus toxoid. And so that's how the results will get spat out. One little tricky bit that often comes up is when we go to the next page and you have to select each well that's an unknown or a blank, we often get a lot of questions about what's our control and what's our standard. And, you know, whilst we refer to our standard curve as a standard curve, that differs from what the machine refers to as a standard. So on the machine, you have 4 options. You can tell it that in this well, it is an unknown, it is a blank, it is a control, or it is a standard. And the machine, when it's referring to a control or a blank, it's referring to the commercial kits that are available for these machines. And in these commercial kits, you are provided with controls and standards to run alongside your assay. Because our COVID assay is comprised of an in-house panel, so a panel that's been designed and developed within a lab and not commercially made, we don't select these. So the only 2 buttons that you'll select when setting up your plate are blanks for your 2 blank wells, and everything else, including your standard curve, is considered an unknown. And you'll know it's your standard from your plate layout, but the machine needs to think of it as an unknown. And then essentially you press run on the machine and you'll start to see a screen like this in the bottom right where things are being classified onto classifications in terms of like the amount that's there and the bead ID region. And so each little blip within this graph or circle of dots represents a different bead region. And so the more bead IDs or antigens you're looking at within your assay, the more you'll have on your graph in terms of number of blips. As we mentioned, the plate will probably take about 60 minutes if you've got a full plate. So you can go away and have your coffee or your lunch. And when you come back, you'll be able to save the results and they should be exported as an Excel document, which is best for the R-Shiny app. And again, just double checking that all the nomenclature on your raw data file is correct and consistent throughout. And so I really can't emphasize enough that you should start as you mean to go on. Ensure that there's no spaces in antigen names. And then when it comes to actually uploading your plate layout for our Shiny app, it's really important that the top corner, so in this A1 dot or A1 cell, that it says plate, and that allows our Shiny app to actually determine that this is P plate, your plate layout, and we'll read it from there. If you don't have plate, the app doesn't work. If you have blanks, as I've suggested, you'll have 2. Make sure they're labeled sequentially, so blank 1 and then 2, so it knows that there are 2 different samples and doesn't merge the 2. And we make sure that our standard curve is labeled S1 to S10. And then if you have all of that, you can upload it into the Our Shiny app and it spits out these beautiful graphs and wonderful results. And I know that Maria and Shazia are going to take you through all of that in more detail. So I'm just going to finish there and say thank you so much for your time. And if you have any questions at all, whether it's in the next week or in 3 months' time when you start to do this, please reach out. My email address is on this slide. And I'd also really encourage you to check out the link on this slide as well. It's part of the Luminex website. It's got a great tab of videos and webinars and a bunch of YouTube videos that are like really helpful in terms of taking the machine out of the box all the way through to troubleshooting your assays. But yes, thank you and happy to take any questions.